In 1952, Alfred Hershey and Martha Chase conducted experiments to prove that DNA is a genetic material. They used bacteriophage as only the genetic material enters the cell when it infects it. The virus, that is bacteriophage, was grown in radioactive sulfur and some were grown in radioactive phosphorus. Viruses that grew in the radioactive sulfur had radio-labeled proteins, as sulfur is not present in the DNA but only in the proteins. Viruses that grew in the radioactive phosphorus had radio-labeled DNA, as phosphorus is present in the DNA and not the protein. The viruses were then allowed to infect bacterium, that is E. coli. Radioactive sulfur remained outside the cell, whereas radioactive phosphorus entered the cell. After centrifugation, the bacterial cells form a pellet, whereas the rest form the supernatant. Radioactivity was found in the supernatant where the sulfur was labeled, whereas radioactive pellet was found where the phosphorus was labeled. Thus, this experiment proved that the genetic material was DNA and not proteins.